right? We have the recordings also. Good morning. And 6 o'clock, see, the time is okay. The light has also fallen, so it is not a difficult thing. And here, actually, I'm not telling you to switch on the video. Boys can switch on the video, right? I'm not telling the girls to switch on the video, right? However, I think everybody can hear me clearly, right? Today we'll discuss that lesson, biology part. Last day we completed the lesson friction along with the practicals. You got the readings and you understood everything. And limiting friction, dynamic friction, and what is that? Static friction, the graph and everything you studied, right? Now today I hope to do the biology part because we have to do the things balanced, right? I mean physics part, chemistry part, and biology part. And this part is actually you are learning so many things regarding the cell and plant cell and the animal cell, the differences of them and the organelles of them. Organelles, I'll tell you the definitions in each and everything, and especially the cell theory, right? About the cell theory, and the cell division is also there. I don't hope to do the cell division part because that a large amount of matter that can't be deposited in your mind, right? Up to a certain extent, will complete, right? Now take out your tube. Take out your tube. That tube is nicely made. I tried my level best to make that nicely, right? We see, right? Take out your tube. The structure and function of the plant and animal cell, right? Open the tube, right? And you can see an animal cell is something that at the cover, I'm telling about the cover. The animal cell, it has got a little bit of the circular shape, and the plant cell, it has got a something like a square or the rectangular shape. There you can see there are various kinds of things embedded in them. We call that the word embedded. Embedded means sunk, right? They are inside the cell, right? And can you see those things from your eyes? No, you can't see them from your eyes. And if you use the normal microscope, compound light microscope, you can see the shape of the cell and you can identify some parts. I mean the nucleus you can identify. Sometimes the vacuole you can see. Sometimes the chloroplast you can see, and the cell, the margin of the cell you can see, the shape of the cell you can see. A part of that you can't see the other organelles, right, from the light microscope. So here we are learning the electron microscopic views of the plant cell and the animal cell, right? Electron microscopic view of the plant cell and the animal cell. Regarding the electron microscopic view, you can identify each and every organelle inside the cell, right? Now suppose that if you look at the human body, right, you think about the human body from the eye, you can't see the cell even, right? You can't see the cell even, you can see the tissue, right? The skin, that epidermal tissue you can see. But inside that, there are so many kinds of cells, they are invisible to our eye. And I told you these kinds of organelles are not visible to the light microscope. You have to use the electron microscope to see them, observe them. Right, so then we will move to the theory part. First, I have asked to write down the definition of the cell. Right, to write down the definition of the cell. Better as share the screen footage with the cube, then you can understand the things to write. The structure and function of the plant. Here. You can see that clearly. Right? Yes, okay. And first you have to, we have to complete these parts, right? First you have to define the cell, right? Then just we'll have a discussion, right? What do you mean by a cell? You have learned that in the small ages. In those grades, what have you learned today? What's the meaning of the word cell? Yes. Can somebody tell me?
The basic structure and functional unit of life.
Yeah, the basic functional and structural and functional unit of life. Actually, that is the definition. Cell, that is the smallest unit regarding the living body, but actually that is not the smallest one because you have learned the atoms are the smallest particle, but that structure, the visible level using any kind of a microscope or something, electron microscope, that is the cell, right? So that is good, that is correct. The structural and functional, right? Structural and functional means that the cells are making the structure. Cells are doing the function, right? There are so many cells, they are doing so many functions. For example, we say that some things you have learned up to now, right? Red blood cells are there, they transport oxygen, right? Bone cells are there, they give the support to the body, right? Then neuron cells are there, they transmit the impulses, transmit the messages throughout the body. So in that way, they are doing various functions. So that is the structural and functional unit of the human body or the living body, right? We'll write the definition to the write down the you write the structural and functional unit of the living body. The structural and functional unit of the living body. So that is the definition of the cell, right? Then we have to learn about the history of the cell, but no need to learn so much things, right? The things they have given in the textbook is enough, okay, right? Then the history of the cell. From the history of the cell, they are giving some ideas that the people have found up to now in the previous ages. No need to keep reminding the years, right? Just the name and their significance, right? The history of life, right? There's a person, the first person is called Robert Hooke, right? He's the person. Now we'll see in the textbook you can find the significance. I mean the foundings of him. Just read and tell me from the textbook. Yes.
He observed cells for the very first time in a piece of cork by using his own light microscope, and he observed a small beehive shell-like structures and named them as cells.
Yes, right. He has done some experiments. Very good, right? He has done some experiments. He had made a microscope by himself, right? We will see that microscope is given below. You can see that, right? Here you can see that. Right? You can see that, and I have given to name those people, those pictures also. And there you can see the second picture. Can you see the second picture? That is the microscope that he has made. He has used his own microscope to identify cells, right? He has used a thin slice of cork. What is that? Thin slice means that, thin slice means you can use a blade and take a thin slice. Slice means the very thin membrane, right? Cork means cork. I think you have seen that in champagne bottles, the, there are some wood stoppers, right? That they are called cork, right? Cork, how it is made? You know that the process of making cork, you have learned in previous pages. Cork, how it is made. You know that there are mangrove plants. Have you seen mangrove plants? In those mangrove plants, you have learned in grade 8, yes, I suppose 8, 7 or 8, right? You have learned respiratory roots. Can you remember respiratory roots? Right, respiratory roots you can see in mangrove areas. Something like the spines coming out of the water, right? Something like thorns, right? That you can see in the mangrove plants. Near the mangrove plants, there are respiratory roots. They are the plants, they use those roots to make cork stoppers, right? Cork stoppers. You can see the champagne bottles, the stopper, and some kinds of honey bottles, they are using that cork stopper to close that. Right? They are somewhat soft, right? They are somewhat soft, and they have taken a slice of that one and observed through his microscope. Then he has seen something like what to think. The cells were seen like beehive chambers of the beehive. Beehive means the beehive. The bees are living in that one beehive. So beehive, they are the shell, the chambers of bees. The chambers of bees somewhat like hexagonal shape, hexagons, right? Hexagonal shapes. Those like structures he has seen, but he hasn't seen the interior side. Just the shape of the cell he has seen somewhat like beehive shape, beehive, right? So better we write that part later, but we have to look at the technical view. You have got the textbook, right? But I have to say it's right. You need, yeah, right? And here you can see the person, Robert Hooke in this picture here. You can see in the screen. That is Robert Hooke. And the second one is his microscope, right? His microscope. And the last one is the cells, a section of the cork. Cells, right? A section of the cork. Name them, Pute. Name them. Right? Name them, then we write about Robert Hooke, right? Write down. Observed a section of a cork. Yeah. Observed a section of a cork. Section of a cork by his microscope. By his microscope. By his microscope. And found structures like and found some structures like found structures like chambers in a beehive. Chambers in a beehive. Chambers in a beehive. And named them as cells. And named them as cells. Observed a section of a cork by his microscope and found some structures like chambers in a beehive and named them as cells. Right? So he is the first person who observed the cells, but you know that just the structure, just the shape, right? Just the shape, and he named them as cells, right? And I have given three people here. The next point. Slidey, Swan, and Radolf Burkhoff. Their names are not, they are not in Sri Lanka. Their names are different from each other, right? Slidey, right? That is one person, Slidey. Next one, Swan, right? Third person is his two words have the two names have given his name. Radolf Burkhoff. Radolf Burkhoff. See it, right? Burkhoff. Chemistry in that way, right? Sliding, Swan, Radar, Burkhoff. There are three people, right? Sliding is one person, Swan is the other, Radar, Burkhoff is the other. So three people have combined together and they have found a special theory, right? That is called the cell theory, right? We will write in the next paragraph. They have given in the textbook. Sliding, Swan, and Radar, Burkhoff introduced the cell theory based on the facts revealed by observing different light tissues through the microscope. Right, then we'll write, they introduced, right now, they introduced the cell theory, they introduced the cell theory, they introduced the cell theory by observing living tissues, by observing living tissues. Right, by observing living tissues. They introduced the cell theory by observing living tissues, right? Now you will think what is the theory? Cell theory, right? Normally we call a theory the word theory. Theory means something like information, right? Information. So they have given some information about the cells, right? Some important points regarding cells as a cell theory. So you have to keep that by hearted. That is actually very easy. They have here only 3 points, right? We'll understand it right now. Completed up to this part. And name the following persons also. First one, Robert Hooke. Second one is microscope. Third one, chambers like cells. The cells, right? So that's enough. Then we move to the next part. Now you have to write the cell theory. Here you have to write the cell theory. What is the cell theory? You have write that. You can write that. No need to write on the screen. You can have got the textbook. The first one, the structural and functional unit of life is the cell. Right? The structural and functional unit of life is the cell. The structural and functional unit of life is the cell. The structural and functional unit of the life is the cell. The main theory, right, that we have previously we discussed the same theory, right? The structural and functional, right? That is making the structures, that is doing the function, so that is the basic unit, right? Then the second one, all organisms are made up of one or more cells, right? All organisms are made up of, all organisms are made up of one or more cells. All organisms are made up of one or more cells. Is that true? Yes, right. Now, up to now, you have learned about microorganisms, amoeba, paramecium. There are so many kinds of microorganisms. You know that they are single cell cell organisms. The others, or most of the organisms, are multicellular. Multi means many. Now, if you consider the humans, the animals, the plants, and everything, living organisms, most of the things they are multicellular. So that is true, right? All are made up of one or more cells, right? That is the second point. Third one, write down. New cells are formed from the pre-existing cells. New cells are formed. New cells are formed from the pre-existing cells. From the pre-existing cells. Right? From the pre-existing cells. What is that? New cells are forming from the previous cells. That is true. Here we are learning about the cell division, right? Now you know that. Now suppose that you have got an injury, right? Injury in the hand. Suppose that. Then after few days, that is cured. That is cured. Then how it is done? Did somebody come and put the cells to cover that? No, right? Then from that place, new cells are growing, right? From that place, new cells are growing and covering that place, right? Regeneration. So that is done by the previous cells, pre-existing cells, right? New cells are formed by previous cells. So that is the cell theory. Now just go through that. And next, on Monday, I'll ask you, right, to tell the cell theory. You just go through that. Then just keep remind. The first one, the structural and functional unit of the living life in the cell. Second one, all organisms are made up of one or more cells. Third one, new cells are new cells are formed from pre-existing cells. Pre-existing means previous, right? Previous cells, right? So that is the cell theory. Then we move to the next one. The cells differ from each other. Yes, that is true. The cells differ from each other by the shape. The shape differs from each other, right? And size, right? Size and the function, right? They are basic knowledge that we have learned in previous grades. Just I gave you to write some examples for this, right? Different types of cells. We will write with the cell and the function, right? Write down first one. Red blood cells. Write down the first one. Red blood cells. Red blood cells. We will write the function in front of that. What is the function? Yes, transport oxygen. Transport oxygen. Transport oxygen. red blood cells. Then we'll write neurons, or neuron cells. Neurons, right? Neurons will write the function what? Transmit impulses. Transmit impulses. I think that word is familiar. Impulses means the messages, actually, right? Transmit impulses. Then the next one will write like blood cells, what else they have, transport oxygen by red blood cells, impulses by neurons, they have given only that one. Then we'll write bond cells, bond cells, bond cells, supporting the body, supporting the body, supporting the body. Then we'll write another one, muscle cells, that is also that supporting the body. Muscle cells supporting the body. And there are so many cells, right? I'm not doing each and everything because up to that part, no need of so many things, right? Okay, that's it. Red blood cells transport oxygen, neurons transmit impulses, those things they have given. Bone cells supporting the body, muscle cells supporting the body, right? And there are epithelial cells, they are covering the surfaces. There are so many kinds of cells, right? Just these things are enough, right? Finish writing. Okay. Then we will move to the structure of the cell, right? Now we will learn about the structure of the cell. We will move to that part. Structure of the cell, right? For that, there's an activity, right? There's an activity, study of animal cells, right? Study of cheek cells. The activity, I'll show you the video. The activity, we'll do that in the school, right? Regarding this lesson, I think 2 or 3 might be 2 or 3 activities are there we have to do. This one is very easy, we'll do in the school, but here we will observe the activity. And I have given an activity for you, that I have given the order of the practical with the, not according to the order, regarding the incorrect order. You have to find that, put the numbers according to the order. Now just observe this one. Study of animal cells. We are observing the cheek cell. Cheek means, you know, the cheek, right? Cheek cell, not from the outside. Open your mouth and inside the cheek, we are observing the cells, right? Then what are the things we need? Sample of cheek cells, glass slide, cover slip, microscope, water, right? Those are the things, right? Wait till you put the video. Just give me few minutes. Just go through the textbook and read the method and try to put the numbers there according to the order, right? I'll show you the activity. Right, that I have given, you have to put the correct number, order, right? You can do that by observing the textbook, right? Structures on the plant cell, I will see whether they have the activity, right? The real activity, you should do that, right, normally. But before that, yeah, they are the practical, I'll show you that. Right, did you put the numbers, right? You can observe the textbook and write them. What is the first one, tell me? What is the first step?
Wash the
Yes.
mouth.
Wash the mouth.
Yes, wash the mouth. Yes, that is to wash the mouth, right? See that if you haven't washed the mouth, you can see the bones and the previous, right, the previous food particles and everything from that microscope, right? So that they are bringing some kinds of germs, right? So that's why they are telling to to wash your mouth first, right? That is the first thing, to wash the mouth. Then tell me the next step.
Scrape the inner side of the cheek using a yogurt spoon.
Yeah, yes, you have to scrape, you have to scrape, right? Scraping means not hardly, right? Taking a yoga spoon and just slowly, you have to take a feel, right? That they are not telling to get something with blood, right? Just now, suppose this is the teeth, right? Just you have to step like this, right? The cells are coming with it. The cells are coming, right? You see the practical one? I don't know whether they will show the way of doing the practical. However, they are showing that the observation, I think, is, no, they are doing that. They are demonstrating. They can share that. Right, here's the activity. Right, can you see that? I hope that you can see this. Right, I will found.
Cheek cells of a person. For this, we will need a light microscope. As you know, this is a compound light microscope because it has two lenses, the objective and the eyepiece. Through that, you can clearly observe the cells of a specimen. And to prepare the sample or the specimen, we need to take cheek cells that you already know. With a very clean washed yogurt spoon, we scrape out a part of the cheek from inside. It's not that we really remove a huge part, just light scraping will give us some tissues. So that will be spread on a slide. As you know, always to prepare a sample or a specimen to observe under the light microscope, we have to use a glass slide. This is something that's familiar to you, and on this glass slide, we need to put a drop of water using a brush like this. We will put a drop of water, and onto that, we will have to spread the cheek cell as a smear. Just spread it on the glass slide. And this, you know, is a smaller, thinner piece of glass which is known as the cover slip. We have to place the cover slip on top of the sample. So here, when we place the cover slip, we don't just put it on top. If we do that, there can be air bubbles trapped inside the sample. So therefore, we have to hold it at an angle and place it on the sample. I have already prepared this slide using a student's cheek cell sample. So I have a temporary slide mount of the cheek cell here. Now we will observe it under the light microscope. So as you know, students, when we are using the light microscope, we have to place the sample on the stage. Here you can see I will place it on the stage, and we have to adjust the sample so that it comes in line with the part where we have the light falling on it, where the lenses are, so that we can observe it clearly. Then I have to observe the sample using the lowest power. Lowest power can be easily identified, that has the shortest tube. And I have already selected the lowest power, and for that the adjustment is done using the coarse adjustment knob. So while I place my eyes and observe the specimen through the eyepieces, I can move the coarse adjustment and observe the specimen in the slide. So now I'm going to do the adjustment. I'm going to move the coarse adjustment knob so that the lens moves. As you can see, I will first bring it down and then I will move it upwards. So now when I move it upwards, you can see, if you can't see clearly, you might have to move it down again. And then when you watch, you can see the cheek cells in that specimen. You can see circular shapes. Different sizes but circular shaped cells in that sample. Once you observe that,
right, I think that's enough, right? That's enough for your knowledge. And you saw that the way of observing that. So, however, you have to prepare the order, Puti. Tell me the order. Yes, tell me the correct order of the sentence, the observation, right? You can observe that in the textbook. First one is wash the mouth. First one is wash the mouth. Then scrape the inner side of the cheek using a yogurt spoon. Inner side of the cheek using a yogurt spoon. Obtain a clean glass and put a drop of water and transfer the specimen onto a slide. Obtain a clean glass slide. Always that purity, that pureness is well concerned. Why? If there's any kind of dirt or something, we can see that. So when you are taking any kind of a thing, you have to clean them well, right? Then clean glass slide and put a drop of water. Always they are telling to put a drop of water. They'll ask why. Yes, why put it? Why they are adding a drop of water? Yes,
easy.
Yes.
For easy visibility.
For easy visibility, but the most important point is that is the thing is to make the specimen live, right? To make the specimen live, right? Without water, what will happen? They are the living cells. They will dry up, right? They will shrink. So it is not live. That so to make that alive, you have to put a drop of water, right? Easily you can see that. But the main point is to make that alive, right? So they are asking some questions in that way, right? Then transfer the specimen onto the slide. Cover the specimen using the cover slip. Now, while using the cover slip in that video, the teacher explained a lot, right? Now, suppose I have got the things, right? I don't know whether you can see this one. This is, you can see, right? This is the slide. This is the slide, glass slide, and cover slip, I think you can't see, Pute. That is a very tiny square shaped one. Now, suppose this is my fingers are the cover slip, right? When you are mounting the cover slip, you have to do this like this, right? Put it an angle and just slowly cover it by the cover slip, right? So that is the way of using the cover slip. The reason is to remove air bubbles. When we are doing the practical in the school, I'll explain you a lot, right? So doing the practical is the best way, right? I'm telling you, right? And I have got this is my microscope, right? I'm using this one to observe the things. And you know that when you are using the microscope, here that has only one adjustment, right? This is not the perfect one. The coarse adjustment is used, right? Not the fine adjustment. Coarse adjustment is used. And normally, if you need to watch a large amount of cells, you have to use which one to take. There are this is the eyepiece. This is the eyepiece. Objective lens. There are 3 types. You know them. What are they? Low power, mid power, and high power. Normally, if you need to observe many cells together, you have to use the low power. If you need to zoom one cell, you have to use the high power. If you need to just to watch moderately, you are using the mid power, right? So this is the way. This is the stage. You know this stage in the school. This is not the school one. In the school also you get this type of a microscope, and we'll observe that. Then you can understand everything, right? So that is the now then cover the specimen using the cover slip without trapping any air bubble. Why? Without trapping any air bubble means if there's an air bubble, we can see the air bubble on it. Something like a black color, big circular one, right? Then and observe through the light microscope, right? Then move to the tube, right? Move to the tube. right? Did you put the numbers, right? After putting the numbers, you have to draw the observation. Draw the observation, not now, Pute, as a homework you do that. In the textbook also you have given, and you saw in the video also the same thing you saw. Then tell me that you saw them as pink color. Then how it has happened? Is the cheek cells pink color? And is the cells pink in color? Hmm? They are not pink in color, Pute. They are colorless, right? Normally the cheek cells are colorless. Then you will think the teacher is telling to take that with blood. No, that is not blood, right? That is colorless, right? To observe them clearly, we can use stains, color stains, right? In the laboratory, there are the stains. I don't know whether they are the color stains in the school. I'll see, right? We have to put a drop of color stain to identify the cells clearly. If the cells are colorless, that you can't observe them clearly. To observe them clearly, you have to use a color stain, right? I have got the colors. I have got them, but now I can't find them. Let me see. Color stains are here. I have got them. Right. So, I try to put the color stains. Here in this set, there are small bottles, right? They have various colors, red color and purple color, right? Colors, green color. We can use the colors, right? We can use the colors and to observe the specimens clearly, right? Okay, you can understand that, right? Then quickly move on to the next part. Right? The next activity is study of plant cells, right? Onion peel, onion cells, right? So for that we need onion peel, watch glass, glass slide, cover slip, paint brush, microscope, water. They are apart from the previous things. They need the paint brush. We'll understand later what the function of the paint brush, right? Method, right? Here also you have to put the numbers according to the order, right? Put the correct order of the numbers according to the steps. Now just go through and complete it, right, by yourself, right? Then while doing that, I'll show you the activity, right? I'll share the activity. Radha, why, what has happened? Are you calling me? It's okay. Right. I was thinking you were in the class and I will call you. Right, then, right, here's the activity. First we'll watch it, right? So far, in the school, you can do the practical easily, right, by observing this one.
Prepare now? Yes. We are going to prepare a temporary slide mount of a plant tissue. So for this, I have chosen the onion. So we'll be taking onion peels and using that we will make slides. And we will observe that slide in a light compound microscope. So as you know already, to prepare a temporary slide, we will need a clean glass slide and a cover slip. And also to cut and take out very thin onion peels, I will need a sharp blade, an onion as the sample or the specimen, and also to initially transfer the small cut out onion peels into water. I have a watch glass with water in it, and to transfer the specimen from water onto the glass slide, I'll be using the brush. So first I will take out a piece of onion, and from that I will cut out a few tiny peels from it. So here we can peel out very thin peels and put it into water. First, I will put it into water. It's better if you can take out more than one peels, so that you can select the thinnest one from the out of all the peeled out parts. So now I have 3 peels. Out of that, I will select one of the peels which is more clear. I think this should do. So first, I'll have to put a drop of water on the glass slide, and onto that, using the brush, I will transfer this peel. I will have to take out this peel. Now I will have to transfer the onion peel onto the glass slide. So now I have kept the peel on the glass slide. Then I will take this cover slip. As you already know, we can't just put it on the specimen, then air bubbles can get trapped. So because of that, I hold the glass slip straight and the cover slip at an angle to the specimen and slowly place it on top of the sample. So now I have my glass slide ready to observe. Then I will have to place it on the light microscope. I have placed the sample that we prepared. Then we will have to first make sure that the specimen is in line with the lenses. So to do that, I can adjust the stage and make sure that the specimen is in the correct place. So by looking at it also, you can adjust it. Then as you know, there are more than one lenses. Those are the objectives with different powers. You all know what power you should use first. Yes, it is the lowest power and that has the shortest length. So with the lowest power, I will have to use the coarse adjustment now. So I will look through the slide and try to move the lens so that I can see the onion peel. Now that I have adjusted the lens, I can see the slight shape of outline of the cells. Here the cells are different from the previous specimen. Here the cells look polygonal in shape. You all also can see it clearly.
That's enough, okay, right? Now we'll move to the tube. And in the tube, you have to complete the order, right? Complete the order with it. What is the first step? Yes, there. I have separated each and everything. And first one, cut an onion and obtain an inner fleshy tissue. That is the first one. Then number 2, remove a peel from inner or outer surface of it and transfer it onto the watch glass. Remove outer surface of it. Then transfer it onto a watch glass containing water. Next step. Then put a water drop onto a clean glass slide and then transfer the specimen onto the slide using a paintbrush. Then cover it with a cover slip without trapping any air bubbles. The last one, observe through the microscope. That's all, right? So did you put the numbers, right? Put the numbers according to the order and draw the cells, not now, as a homework, right? For the observation, you have to draw the cells, right? In the practical, it was clearly seen. I think you could see that clearly. The pink color, the cells, polygonal shape, right? So that is it, right? Then we move to the next one, right? Then we are starting the most important part, right? That is the typical cell, right? That is the typical cell. Okay, the battery is a little bit low. That I'm sitting off my video and you can see the screen on my head, right? And the first one is the typical cell, right? Typical cells. So questions they are asking what is the meaning of the word typical cell, right? The normal word typical means that the no need of that normal word, right? Just tell me, have you found the answer of the word typical, right?
The cell prepared by including all the organelles is known as the typical cell.
Yeah, that is true. Very good, right? Typical cell means that is a kind of a cell that is represented all the organelles, right? Now, see, will you in this picture that I have just added from the internet, right? A plant cell, the structure of the plant cell, right? It has all the organelles. Now, see, many organelles are there in that one. But if you take the cells, the natural plant cells, all the plant cells do not have every organelle, right? That may be some organelles may be excessive, some may be lesser, right? Some may be absent, some may be present. But here we have made a cell including all the organelles. Why do we use that typical cell?
Studying, easy
Yes, to
study.
study. Yes, actually to study, right? But naturally, these types of cells are not existing, right? These types of cells are not existing because some are having not this shape, some are having greater amount of mitochondria, some are having less amount of chloroplast, so not the same thing, right? So for the ease of study, they have given a cell including all the organelles, right? So that is known as the typical cell. Write down the answer. The cell prepared by, write down here, define, there, write down. The cell prepared by, the cell prepared by, including all the organelles, including all the organelles. How do you write organelles? organelles, including all the organelles. I am telling the things from the page number 112, the last paragraph. Including all the organelles. That's all, right? The cell prepared by including all the organelles. Once that question was there in the paper, what is the meaning? What is a typical cell? So that is the answer. The cell prepared by including all the organelles, right? Then I have asked another question. What are organelles? Can you see the screen, Pute? Cute? Yes, Asa, can you see the tube in the screen?
Yes, teacher.
Yes, right? Okay, that okay. When it is not visible, just tell me. Sometimes they are not sharing. When I share, it is not sharing in the Zoom app, right? Easy. Then what are organelles, right? Then how do you explain that? What are organelles? That is in the first line of the typical cell. To define that, the small structures present. Yes, the small structures present within the cell to perform different functions. Did you find that? Page number 112, typical cell, the topic, the first line. Write down the answer. The small structures present within the cell to perform different functions. Different functions. You will understand the functions of each and every organelle. Today we are learning. We will draw the cell, but I'm doing that as a homework for drawing because the time is not enough for us to draw and learn the things. That is for a homework. I have left the space to draw them, but each and every organelle we will study, right? Did you write the answer, Pute? The small structures present within the cell. You can see that here. Within the cell, you can see so many small structures, right? Within the cell, to perform different functions. They are doing different functions. The nucleus is doing one function, ribosomes are doing one function, Golgi body, they are doing one function, mitochondria. That words are not familiar for you, I know that, right? But when you read them again and again, the words will be very familiar for you, right? Vacuole, chloroplast, smooth endoplasmic reticulum, that peroxisomes don't end up on those things, right? Some few things are given in your syllabus, that learning that much is enough, right? Then in the first day you can't tell each and everything, right? You have to practice it, right? After practicing only, you can tell these things as me, right? Because I have taught so many years, so the words are familiar for me. And you also practice again and again, then you will tell all the things clearly, right? So you can see the plant cell, the structure I have been, that is taken from the internet, just to see that, right? Then draw and label the parts of the plant cell, right? Draw and label the parts of the plant cell. You can see that in your textbook, right? You have to go through that, and by don't get the photocopy and paste. I'll check that, right? You have to draw by yourself, right? If you can't draw, it is easy to just draw with it, right? Draw with it. And if it is unable to draw from your free hand, use normally the half sheets. Nowadays the half sheets is like tissue papers, right? You can see through them, right? You just keep the half sheet on the book and draw the outlines, and then draw the organelles and label the parts. Here you have to then paste it here, right? Don't get the photocopies and paste here. I'm telling that definitely don't get the photocopies. Draw by your free hand, right? Draw and label the parts. So these are the things. Drawing things are for your homework, because you have 3 holidays. In those days, in these holidays, you can draw them. I'm not telling you to write something, right? Draw and label the parts, right? So in the textbook picture, better to share that. No, I think you all of you have the textbooks, right? So no, I'll just from this one I'll explain. You just watch your textbook also, right? They have given the outer line, right? The outer line. What is the name of that outer wall-like structure, Puti? What is the name given? Wall-like structure. Cell wall. Yes, that is called the cell wall. Cell wall is something you just think this like the house, right? The wall of the house. So the same as cell wall, right? We'll observe the videos animations. Before that, I have to explain it, right? Cell wall. Then after the cell wall, the outline, right? I point here. You just watch your textbook. Here, there's another line. Here. After the cell wall, that line is called the cell membrane, right? That is called the cell membrane. Did you find that in your textbook? No, they have done plasma membrane, right? The cell membrane is the same. Plasma membrane. So outer layer is the cell wall. Then comes cell membrane, plasma membrane. Then the medium. What is the medium? The name of the medium. Did you find the name of that medium?
Cytoplasm?
Yes, that is called cytoplasm. Have they given with a cytoplasm? Golgi complex, plasma membrane. They haven't marked in the textbook. Am I correct?
Yes, in the textbook we don't have, no.
Yes, we don't have in the textbook, but better you label them, right? The medium is called cytoplasm. So in this you can see that I circled now cytoplasm. You name that, right? That is cytoplasm. And at the middle, there's a light blue color, large one that is called the vacuole. Here, when I'm telling, I'm circling them, vacuole, right? That is the vacuole. In your textbook also, you can see the vacuole. In the vacuole, they have named the parts. In the textbook, cell sap and tonoplast. Did you see that, Pute? Cell sap and tonoplast. Cell sap means the liquid matter inside here. Here, that I am putting the dots here. This is the cell sap, right? Liquid part. The outline is called the tonoplast, right? Tonoplast. Read them again and again and practice them, right? Then I'll tell you, right? Take a paper, draw the plant cell and label the parts. So that you should have that ability, right? Practice them there. Right? Cell sap and tonoplast, totally we call that the vacuole, right? Then there's a very prominent purple color one in the screen you can see. In the textbook, in the side you can see a ball-like structure that is called the nucleus. Can you see this one? Nucleus. I squared it. Nucleus, right? In the nucleus, they have named three parts. What are they? Two things. Nuclear envelope. Nuclear envelope. Envelope means the cover. Envelope, normally we use the envelope as a cover. So envelope is the cover of the nucleus. And nucleolus. Actually, I covered that nucleolus. Sorry. Right? That this part, that tiny middle part, that is called the nucleolus. Right? Then all together, this structure that is called the nucleus. Right? That is called the nucleus. Then in the textbook you can observe that. Then there's an orange beautiful part here. These are mitochondria. Yes, we call them mitochondria. Mitochondria, right? They are called mitochondria. In the school book also you can see mitochondria, right? Mitochondria. Mitochondria means mitochondria. Then mitochondria. And there are green color parts here. These are green color things, the same shape of the mitochondria but green in color. They are called chloroplast, right? Chloroplast, green color pigments, not the pigments actually, organelles, they are chloroplast. Inside there you can find chlorophyll, right? Chloroplast, green color thing. And yes, those are then something like stack-like things, right? That purple, pink color thing put here in this screen, pink color, we call them Golgi bodies, right? Golgi complex. Golgi complex. We call that here. I'm squaring now. Golgi complex. They have given Golgi vesicles, but the word that you have given in your textbook is Golgi complex, right? Golgi complex. That words are very nice, right? Golgi complex. And there are tiny dot-like structures in your textbook, right? Tiny dot-like structures. I removed these things then you can see them clearly, right? Here also you can see the dot-like structures. Here dot-like structures. They are called ribosomes, right? Ribosomes. Ribosomes. They are called ribosomes, right? Ribosomes. Then apart from that, we see whether we have. Then there's another thing. In the nucleus, the outer cover, there are some things here. I'll put an arrow here now, right? You can't see them clearly. There are dots connected to these parts. Dots connected, right? So we call them rough endoplasmic reticulum. Rough endoplasmic reticulum. In short form, we say RER, right? We say rough as there are dots around them. Because connecting those dots, the dots are the ribosomes actually, right? After connecting those dots, the surfaces become rough. So we call them RER, rough endoplasmic reticulum, right? And they are smooth endoplasmic reticulums. So they haven't given. But there are some other things here, this part I'm circling. That is smooth endoplasmic reticulum. We say SER, right? We say SER. Smooth endoplasmic reticulum. Why do we say smooth? Smooth means they don't have that dots, right? That's why we consider that as SER. Smooth endoplasmic reticulum. Right? Just I'm labeling the parts, right? Then after that I'll teach you about each and every organelle, right? Then is that all the things that given in the textbook? We'll see. Ribosomes I taught you. RER taught you. Chloroplast, yes. Cell wall, yes. Mitochondrion, yes. Vacuole I taught you. Cell septum and the tonoplast. Golgi complex, correct. Plasma membrane I told you. Nucleus I told you, correct, right? Then apart from that, there are many, right? But those parts are enough for your language, right? Your knowledge, right? So your knowledge, right? Then that part is over, right? Okay. Now, then you have to draw and label the parts of that picture, right? Then I have given the animal. Before that, we'll watch a video, Putin. We'll watch a video regarding the plant cell, right? Plant cell. Right, we'll watch an animation. It will get only 2 minutes, 3 minutes, right? I'm not showing you lengthy things, right? I don't like that. Not much of things, right? That is very nice, right? This lesson is very nice. Just focus your mind. Morning time is a very good time for you to keep your mind. I don't know whether you can understand this method, but actually, learning online is not something bad, right? But the thing is that the parents also should know these things, and basically you should understand this one, right? That learning using the devices, that is not a bad thing, right? The way you handle the instruments are the bad things, right? Don't use them to do the games and do the other kinds of bad things. Don't use the sources, right? These are actually, I mean, the knowledge houses, right? So you can get so much knowledge from these devices, but don't misuse them. Don't misuse them. You get the knowledge and be honest to yourself. Always be honest to yourself. Then only you can do the things well. If your mind says don't do these things, do the bad things, that you are not honest and you are going in the wrong way, right? So there's no enough time for you to sit for the O-level, and your target should be getting 9 days. Everybody, right? I'm telling everybody to get 9 days. So try hard. I'm always telling you can. I have never told you that you can't do this. I'm always telling you can do this. So focus your mind on studies and always try hard to study and show your talents in the test, right? So then only you can go forward. Because nowadays, without knowledge, nobody can go forward, right? So we'll see this one. And you know that this time, A-level results, the island first, island second, island third, island fourth, most of them have studied online, right? So online studying is not a bad thing. You can get the knowledge there. But the thing is, the students don't know how to use the resources, right? When they go in wrong ways, the parents don't like to give those devices for you, right? You are not behaving well, right? So that is true. But if you are doing the things well, that is a good source of learning, right? Now, can you see the screen, Putin? Yes, can you see the screen? I'm going to play the video. Can you see the screen? Everybody tell me.
Yes, teacher.
Yes, right, okay. Right, here's the video. Just go through the video and just listen there.
A plant cell, similarly to an animal cell, contains a cell nucleus, in which genetic material is stored in the form of DNA. Inside the cell nucleus there is the nucleolus. It is a granular body built of protein and ribonucleic acid, RNA, in which, inter alia, ribosomes are produced. Ribosomes are organelles involved in the synthesis of proteins. Some of them are attached to the endoplasmic reticulum. Others are freely suspended in the cytoplasm. The cytoplasm is a liquid suspension filling the cell, which contains various organelles. One of them is the endoplasmic reticulum. It has the shape of a labyrinth surrounding the nucleus. One part of the ER is called rough, and the other smooth endoplasmic reticulum. The Golgi body is the cell repository. The substances produced in it are stored there and then transported to other parts of the cell. Among the more complex organelles are the mitochondria, in which cellular respiration takes place. Their number, size and shape vary depending on the cell type. Each plant cell is surrounded by a cell membrane. It acts as a barrier separating the cell from the external environment. A plant cell also has a number of unique features. The cell wall, which is composed of multiple cellulose layers, is a characteristic feature of a plant cell. Its rigid structure serves as a frame that gives the cell a regular shape. The cell wall protects the cell against water loss, while allowing the flow of various substances from and into the cell. The plant cell can use solar energy as fuel for the production of the organic compounds it needs. This is possible through chloroplasts found in the cell, which are also known as green bodies. These organelles contain chlorophyll, the green pigment enabling the conversion of solar energy into the energy of chemical bonds. Large vacuoles, whose surface often occupies almost the entire cell, are another unique feature of plant cells. Vacuoles can be likened to huge tanks filled with cell sap, containing mainly water and spare substances. These elements of the plant cell closely cooperate together, ensuring the proper functioning of the cell. They are found in all plant cells, irrespective of the type of plant.
Right here, right, okay. Right, that's it, right? That is regarding the plant cell. And you saw the particles. You can see more there. You can see them, right? Then we'll move to the tube, right? That is the animal cell, right? Animal cell. Regarding the animal cell, in this picture, now you are a little bit familiar with the words. Here also they ask the nucleus, they ask the nucleolus, RER is there, rough endoplasmic reticulum, and chromatin. Chromatin, did you hear in that video footage that thread-like structures inside the nucleus, right? And centrosome, you don't need to learn that, right? No need of the centrosome. And Golgi apparatus means Golgi complex and cytoplasm. Small vacuoles may be. Normally, animal cells, we can't find the vacuoles, right? And the other things on it. And smooth endoplasmic reticulum, SCR, that is, you need them, right? Now, just go through the textbook picture, Pute. What are the textbook pictures? Pute, wait till the tractor is going so you can't hear the things. Wait till it goes, okay? Right. Right now, look at the picture in your textbook. They have given SCR, right? Smooth endoplasmic reticulum. And cytoplasm, you know, that is the medium. Ribosomes, you know, they are tiny ball-like structures. Golgi complex is also, you know, that is something like a stacking on each other, right? Something like a sandwich, right? Then centriole, they are here. The centriole is, you have to learn there. Centriole and centrosome, the same, right? Centriole. And plasma membrane, you know that. Nucleus, you know, nucleolus and nuclear envelope is there. RER is there. Mitochondrion is there. So those are the things, right? Those are the things. And then then there's something for you, right? That is to label the parts here, draw and label the parts. Then you have to write the drawing and label the parts. You have to do that at home, right? Write the differences between the plant cell and the animal cell. Before that, we'll watch the animal cell also, right? Right, 7 animal cell, right? 7 minutes very time. 7 කමක් නැහැ නේද? 7 බලමු අපි ඒක forward කරන්න පුළුවන්ද කියලා බලමු. ඒකේ පුරා හොඳට ඒක explain කරනවා cell එකට. right? 7 minutes මොකක්ද මේ අපි කොච්චර time waste කරනවා. Right, you watch this one today.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use.
You just get an idea, but simply
The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes.
10 minutes of it.
Some organisms, such as plants, that are
Are you there?
photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens.
See, that
It's
is very
green
beautiful.
because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall.
That is the difference.
There are many other unique structures that only some cells have. Here are just a few. In humans, for example,
See, that's
the
enough,
respiratory
right? That's enough. But now you saw the organelles and their functions. Now you have something to do, right? You see, in the tube, I'll share the tube, right? In the tube, you have to write the differences between the plant cell and animal cell. You saw in the video, right? I think you saw in the video. You can write these things. Write them in the textbook also. They have given page number 114. Animal cell, you can write no cell wall. We write in our way, right? I don't no cell wall. Right here. Right here, write down no cell wall. No cell wall. The other side, write down present a cell wall. Right then. And here regarding animal cell, you can write large content is cytoplasm, right? Large content is cytoplasm. Large content is cytoplasm. Cytoplasm means the medium, right? It has a large medium. Regarding plant cell, they are telling cytoplasm is pushed peripherally. I'll teach you. Cytoplasm, writing cytoplasm is pushed peripherally. What do you mean by peripheral?
To a corner.
Yes, to a corner. Very good. To a corner. Peripherally means to a side. Why? Plant cell, you know that Pute, there's a large vacuole, right? There's a large vacuole, so there's no space at the middle, right? Due to that large vacuole. So that has pushed peripherally to a side, right? Then the next one they have given no, you write here no large vacuole. Yeah, write down no large vacuole. No large vacuole, but they are telling sometimes few small vacuoles may present. They inside brackets write down. Sometimes few may present. Sometimes few, that's enough. Inside brackets write down. Sometimes few. In short form, you write now. No large vacuole, sometimes few. Right? Then regarding plant cell, we will write. Regarding plant cell, we will write a large central vacuole. A large central vacuole. A large central vacuole or few vacuoles. Or few vacuoles. Or few vacuoles. Right? A large central vacuole or few vacuoles. The last one, no chloroplasts. Right? We don't need chloroplasts. Right? Write down. No chloroplasts. No chloroplasts. The other side write down. Chloroplast present. Chloroplast present. Chloroplast present. Right? So those are the differences. Animal cell, no cell wall, plant cell, present the cell wall or with the cell wall. And animal cell large content is cytoplasm. Plant cell cytoplasm is pushed peripherally. Animal cell no large vacuole, sometimes maybe, right? Plant cell a large central vacuole or few vacuoles. Animal cell no chloroplast. Plant cell chloroplast present, right? Okay, now clear? Is that clear? Now today I hope to tell you the functions of those parts. From that we'll conclude the lesson, right? Only the functions and special things, right? Now what's the time? 7:22. By 7:45 we can complete. Before that also, right, we'll see. Right? Is that very tired? No, not tired. Why you feel tired? Right? Teachers feel tired. Always talking. Right? Right? We feel not tired. Now we are at home in a very free mind, relaxed mind. All right, we'll see. Now I have given you a table. You categorize the things there. You put the things here. Then that will be easy for you to do the studies, right? First, we'll write the things regarding the cell wall, right? First, we'll read each and everything in the textbook. Then we'll summarize them, right? Regarding the cell wall, they have given the outermost covering of the plant cell is the cell wall. It is a dead structure. The main constituent of it is cellulose. The main functions of cell wall are to maintain the shape of the cell, support and protection of the cell, right? So those are the things. I'll give you the note, right? Write down the first one, outermost covering. Here write down outermost covering. Outermost covering, right? Then next point write down dead. Dead. That's enough, right? This is the kind of a short now. Dead. Then made up of cellulose. Made up of cellulose. Made up of cellulose. That's all regarding that side. Then functions we will write. Maintain the shape of the cell. Maintain the shape. That's enough. The cell we know that. Maintain the shape. Then we will write shape of the cell. Support. Next one, support. Last one, protection. See how easy these things to study well and protection, right? That is the things regarding cell wall, right? Then we'll move to the plasma membrane or the cell membrane. Plasma membrane, the cell membrane, the same, right? We'll read plasma membrane is the present interior to the cell wall of the plant cells. When we consider about the plant cell, that is interior to the cell wall. But when we consider the animal cell, that is the outer cover, right? You know that. Outer cover, plasma membrane. Can you hear me? Yes. You can. Right? If something happens, just inform me, right? I'm just talking, then you have to tell if something happens, right? If you can't see the screen now, if a kind of a problem happens, tell me. Then the boundary of the animal cell is the plasma membrane, right? It is made up of phospholipids and proteins. Phospholipids and proteins, I think you have learned in the first unit. Making structures, the proteins, making structures, there you have given plasma membrane, right? In the first unit, the functions regarding the proteins and lipids, you have given those things, right? Phospholipids and proteins. Then they are telling plasma membrane is a semi-permeable membrane. What do you mean by that word? Semi-permeable means they select the things and let them to go in or go out, right? They select the things, not all the things, right? When all the things are going through that, we say that is permeable. Semi-permeable means they select the things and they let the things to go in and they let the things to go out, right? Just like the school, everybody can't come, the selected persons can come. So in that way, that is semi-permeable, right? The main function is it is to enclose the cell, that is covering the cell, allow entry of water. Now see below the things they allow to enter water, ions, some molecules, right? And thereby control the entry of existing materials into and out of the cell. Plasma membrane is also known as the cell membrane. The plasma membrane, here you can write to the cell membrane. Here write down cell membrane. Cell membrane. cell membrane. Then we'll write here the special facts. We'll write plant cell interior to the cell wall. Write down plant cell interior to the cell wall. Interior to the plant cell interior to the cell wall. Then write down animal cell, the boundary. Then made up of phospholipids and proteins. Next one right now. Made up of phospholipids and proteins. Made up of phospholipids and proteins. Semi-permeable membrane. You can write in because I have no space. You write even using a large font size. You can write, right? Semi-permeable membrane. Semi-permeable membrane. Semi-permeable. Right. That's all regarding special facts. Here regarding functions, write down enclose the cell. Enclose the cell. Enclose the cell. Then what allow entry of water, iron, some molecules. Allow entry of water, ions, some molecules. Water, ions, some molecules. Control entry and exist. We'll write in that way. Control entry and exist. Control entry and exist. That's all, right? So those are the things regarding plasma membrane, right? The next one, we'll write about cytoplasm, right? Cytoplasm we will discuss. The gelatinous lipid part of the cell, including organelles, is known as the cytoplasm. Inorganic and organic substances are present in it. The function of the cytoplasm are to maintain the shape of the cell. They are the organelles and carry out metabolic processes. The structures submerged in the cytoplasm are named as organelles. Some organelles are surrounded by cell membrane. That means this you know it, right? We'll write gelatinous liquid part, right? Cytoplasm is considered as a gelatinous, Gelatinous, gelatinous liquid part. Gelatinous liquid part. Having inorganic and organic substances. Next one. Having inorganic and organic substances. You know about organic and inorganic substances, the meaning with carbon and without carbon. Just understand that's enough. Right? Then functions we will write. Maintain the shape. The other side. Maintain the shape. Maintain the shape. They are cell organelles. They are cell organelles. They are cell organelles. Carry out metabolic processes. What is the meaning of that? Carry out metabolic processes. Carry out metabolic processes. Carry
Cell
out metabolic processes. Yes, what do you mean by that?
cell functions.
Yeah, cell functions actually the chemical reactions, right? There are chemical reactions which form complex things. There are some chemical reactions which the complex things are broken into simpler things. So altogether we consider them as metabolic processes. You learn that later, the metabolic reactions, right? So that's it. Now, and they have given this one. Some organelles are surrounded by cell membranes, right? After learning these things, I'll give you the special organelles which don't have cell membranes, but here, when you are writing these things, we'll write them, right? No need to add this part, right? Cell membrane and cytoplasm, those things, right? Then we'll move to, is that okay, Puti? Finish writing? And we'll complete this table, then we'll finish the session, right? I'm not teaching too much. That's enough. Then cell growth and cell division, we will learn that later. Finished, right? Okay, then shall I erase these things? Maintain the shape. They are now. No matter that I'll give you the recordings. If you have missed the parts, you can go through the recording and complete it. Right. Then we'll move to the how many organelles are there. These are actually easy, right? Or up to this part only today, right? Then we'll learn about nucleus, right? Nucleus. We'll read the things regarding nucleus. Nucleus is the main organelle in the cell. It is surrounded by a nuclear envelope. One or two nucleolus and the chromatin body are present inside. I think in the videos you saw the tiny ball-like structures. Sometimes one nucleolus, sometimes there may be many, right? That's why they are telling that. And chromatin body, did you see that thread-like structures? We'll learn about chromatin body later regarding the cell division again, right? Chromatin body are present inside the nucleus. During cell division, the chromatin body converts into chromosomes. The function of chromosomes are to store of genetic materials and transfer inherited characteristics from generation to generation. I am not giving to write these things here, Pute, because regarding the cell division, I'll give you about the chromosomes and everything, but we'll write the things we need here only, right? The number of chromosomes is specific to the species, no need to learn here. I'll teach you again. But here we are learning only the structure and the function. Right here we write covered by a nuclear envelope. Write down. Covered by a nuclear envelope. Covered by a nuclear envelope. One or two nucleolus present. One or two nucleolus present. Nucleolus present. Chromatin body present. We'll learn about them later, right? Chromatin body present. That's enough. Then for the functions, we'll write store and transfer inherited characters. Store and transfer is the other side, write down. Store and transfer inherited characters from generation to the next, from generation to generation. From generation to generation. But this is the function of, this is not the function of nucleus today. Actually, this is the function of chromosomes. So inside brackets, write down chromosomes. Actually, the chromosomes are inside the nucleus. So that is also kind of a function. But the main function of the nucleus, we write next, right? The next main function, control of life activities of the cell. Write down. Control of of life activities of the cell. Control the life activities of the cell, right? So that is it, right? Then we will study about mitochondria. I think you saw those things in the video. Regarding mitochondria, after that, there are very few things we have to write. It is an oval or rod-shaped membrane bounded organelle. Aerobic respiration reactions occur. Respiration is happening inside the mitochondria, right? Respiration occurs. Cellular respiration occurs, takes place within the mitochondria to release energy. So mitochondria is considered as the powerhouse. Mitochondria is the powerhouse, right? Generating power. You know that respiration means the production of energy, right? So it is known as the powerhouse of the cell. The energy produced within the mitochondria is used for the metabolic activities of the cell. Right? We'll write the special facts. We'll write oval or rod shaped. First one write down oval or rod shaped. Oval or rod shaped. Next one write down. Actually, they have two membranes. We'll write this one double membrane. Double membrane. That is not given in the book, but we have to write double membrane. So that is enough. Then the other side write down powerhouse. Powerhouse. Next point write down aerobic respiratory reactions occur. Aerobic means with oxygen. Aerobic respiratory reactions occur. Aerobic respiratory reactions occur. That's all, right? That's all. Then Golgi complex completely we complete these are very easy things, right? Golgi complex, one membrane, that means covered by one membrane, right? One membrane stacked on top of the other. Stacks, can you remember, just like a sandwich, right? Stacks, stacked, stacked on one another. On one another. With secretory vesicles. With secretory vesicles. How do you write vesicle? V-E-S-I-C-L-E. V-E-S-I-C-I-L-E. Right? Vesicle. Now, did you remind Pute that regarding the Golgi complex in that video, small balls are separating and going somewhere, right? After learning this one, watch the videos again, right? I'll send you the recording. Secretory vesicle, right? Then we'll write the functions. Production of secretory substances. Write down. Production of secretory substances. How many days? Substances. Production of secretory substances. Packaging. Next one. Secretion. Last one. Packaging. Packaging. Last one. Secretion. You can understand. Producing secretory substances, packing and secreting. Just like the post office, right? They are taking from one place and they are going to the other place and delivering them, right? So that is regarding the Golgi complex. Uday, better we'll discuss the other things the next day, right? There are some things to remind. Now the time is enough because one and more than half hour, everything can't be stored in your mind, right? So that will be enough for today. right? Now go through these things, and on Monday we'll learn the other things. You just inform me whether this method is good or bad. If this is good, we can do some other lessons. I mean, not all the lessons. We can cover the syllabus quickly along with the school and by this method. But I don't have much time today because I have so many classes. When I have the free time early in the morning, in some days we can do the things like this, right? Then previously I can inform you. Then but you should have a tutor also. Without a tutor, we can't go in this method. If it is okay, if you can understand the things in this way, we'll move in this way regarding biology parts. Biology parts, that will be good, better, right? But chemistry and physics, better to learn in the school. But biology parts, you can observe these things in this way. But just you tell me your idea on Monday, right? And so that's enough for today. And go through the things well. I'll send you the recording, watch them again, and learn the things well, right? Up to the cell division, I'm adding the parts regarding the things that we have learned in the term test paper also. So go through them well, right? Okay, then you will stop the class. Right,
Excuse me, teacher.
yes, sir. Mother?
Up to where do we have for exams in this subject?
That up to that you didn't come that day. Last day I told in the school, right? We completed the lesson, the matter, the lesson, the valencies, we wrote the chemical formula and everything. We completed that lesson, right? Then we completed the friction lesson with the practicals. Along with the practicals, we completed last day we did 3 periods, right? And then this lesson up to this part, and we didn't get the timetable yet, right? So I try to complete the remaining part, and also I try to complete the Newton's laws lesson, right? If that is possible, that will be included, right? Okay.
Okay, teacher.
I learn the things that I have taught you. So that is the first term units. Altogether, those are the collection. I'll try to my level best to complete them, right? Right, then we'll stop, right? Then how do you say? I told you how to stop the class in the school. Yes, you can
Thank you,
stand up.
teacher.
Yes,
Thank you, teacher.
that is
Thank you, teacher.
right.